Hi, everyone. This is Karen Huffman, and I think we'll get started here. I just wanted to welcome everyone to our seminar. As you can see from the slide, it's the overview of analytical services here at Pion. I am the marketing manager, and I also take care of our distributors throughout parts of the world. I'm going to introduce our presenter in just a minute, but I wanted to go over a couple things on how this is going to run today. So everyone will be muted throughout the seminar. You can write any questions during the presentation and throughout the Q&A. If I could just ask you to remember to use the Q&A tab down on the bottom of your screen. You'll see a Q&A tab and a chat tab. But if you use the Q&A tab, then after the seminar, I'm going to get a report with all of the questions, and that way anything that we couldn't, that we ran out of time, etc., all the questions we're going to answer in writing and send those to you as well after the presentation. You will also get a link to the presentation and the recording a couple days following. In our first seminar, a lot of companies' firewalls either sent links into spam or blocked them altogether. So your Pion representative is also going to follow up with you just to make sure you were able to receive it, answer any questions, and then also send you the written questions that I had just talked about. So we will do the Q&A at the end, and Rebecca will be joined by members of her analytical services team, Andrew Kennedy, Amy Knight, Sam Lee, and Carl Box, our Chief Technical Officer for Europe, will also be on board to answer questions. So with that, I just want to introduce you to Dr. Rebeca Ruiz. Rebecca obtained her European PhD in analytical chemistry from the University of Barcelona in Spain. Her thesis and master's were focused mainly in pKa analysis, studying a diversity of techniques, media, different calculation approaches, but lipophilicity and solubility also formed part of her doctorate. She joined Pion in 2009, which at the time, that part of the business was Sciss, and then we've merged a few years ago. So Rebecca has been with us for quite some time, and she manages our analytical services team. And so now with that, I'm going to turn it over to Rebecca.
Thank you, Karen, for this introduction. So welcome to this webinar. My name, as Karen says, is Rebeca Ruiz. I am the Global Manager of Analytical Services at Pion. Today, I'm going to talk about what we do in analytical services, who we are, the analysis that we offer, and how to contact us. So, Pion Analytical Services Centre of Excellence is based in Forest Road in UK. We determine physical-chemical properties, absorption, permeability techniques, dissolution, and biopharma. During this presentation, I will describe some of the services that we offer and show selected case studies based on real samples. So let's start. During my PhD at the University of Barcelona, I studied ionization constants. At that time, I was using very conventional potentiometric technique, showing on this slide. An automatic burette with a potentiometer, temperature was kept constant using a bath with a thermostat. Samples were titrated under nitrogen, and data was collected using Microsoft DOS operating system. So when the first instrument from Pion, formerly Sirius Analytical, arrived at the university, it was a very big improvement. The PCA added automatically the titrants and the media, water, co-solvent mixtures, and octanol. So it measures not only pKa, also the log P. It was provided with a printer on the top of the instrument to get the printouts, and the data could be refined in the same instrument or transferring with a floppy disk to a computer provided with the software pKa log P. Since the PCA 101, several generations of potentiometers were designed until finally arrived the Pion Series D3. The Pion Series D3 is fully automated, performing measurements of pKa, log P, and solubility. Since then, with the amalgamation of Pion and Sirius, the Pion instrument family has increased offering a range of instruments dedicated to measure different properties. But this webinar is not specifically about Pion instruments. This webinar is about what we can do with them in analytical services and about the different types of assays that we are offering. During the preclinical drug development, the number of compounds decreases from hundreds or thousands to the one which entered to the clinical trials. To understand the reasons for candidate failure, there has to be considerable interest in understanding and optimizing the physical chemical properties such as ionization, lipophilicity, and solubility as the key to develop a successful pharmaceutical active ingredient into a drug. For a compound to reach a tissue, it usually must be taken into the bloodstream before the compound breaks down to metabolites and it's removed from the body via excretion. For example, following oral administration during passage along the gastrointestinal tract, a drug experiences various factors such as pH, membrane surface area, transit time, fed and fasted states. All of them may impact on the absorption process. This slide shows the interrelationship between drug physical chemical properties and drug absorption. Drug first needs to be dissolved before they can reach the bloodstream. Only when drugs are in solution, they can be absorbed. Charge state or ionization properties influence dissolution, solubility, and lipophilicity, where only unionized drugs molecules are able to cross membranes. Therefore, the best candidates will be drugs that have been optimized against all these properties. We are specialized in the measurement of these physical chemical properties. Most of the drugs that we analyze are small molecules or drug candidates sent by customers in the pharmaceutical industry. We are experts in measuring pKa, log P, log D, and solubility. However, we also measure dissolution and supersaturation assays, and we have an extended offer for absorption permeability analysis. Additionally, we offer in vitro post-injection behavior analysis for subcutaneous injected formulations. Each analysis is offered at different levels depending on the complexity of the experiment. Screening assays are designed for customers in drug discovery. These assays are high throughput analysis and they are used to study large batches of compounds with no repeats or any adjustment of the conditions. Next level is our gold standard analysis, which offer more detailed studies to assure a result. Platinum level provides the highest detail with optimization of conditions to achieve the best results. Alternatively, we will engage in consultative discussion with customers and agree as a scope of work in advance of starting any studies. In addition to PhysChem analysis, we have an extensive range of analysis under screening, gold standard, and platinum standard umbrellas. Unfortunately, I cannot cover all of them in this webinar. However, I'm going to introduce some case study, so at the end of this webinar, you will have a good idea about what we are doing in analytical service and what we can offer. So let's start with physical chemical properties. pKa and potentiometric log P and solubility is determined using Pion Series D3. We are using 4 milliliter vials and 1.5 milliliters of media is added to the vial. The measurement cell is provided with a spectroscopy UV probe, a temperature sensor, capillaries to dispense titrants and the media. A pH electrode and a stirrer is also included, and the experiment is performed under argon conditions. And this is our first case study, ionization constant measurements. We can determine pKa's using two techniques, the spectrometric and the potentiometric, both using the Pion Series D3. The spectrometric technique requires the drug molecule to have a UV chromophore, and is a sensitive technique requiring only 5 microliters of 10 millimolar DMSO stock solution. pKa's below 2 and about 12 can be determined. The potentiometric pKa method can measure pKa's between 2 and 12 and doesn't require that the sample is UV active. Many drugs are small molecules with only 1, 2, or 3 pKa's. However, we sometimes receive big molecules like the peptide shown in this slide. The molecular weight is around 4,000 and it has 14 pKa's. For large molecules, we can display the regions of ionization and estimate the isoelectric point. The isoelectric pH is when the molecule is completely neutral and often when the drug has the lowest solubility. Like pKa's, two techniques can be used for the determination of log P, log D, the spectrometric and the potentiometric technique. For the spectrometric technique, we use the check flash methodology with quantification using HPLC, UPLC, and for the potentiometric assay, we use the Pion Series T3. The spectrometric techniques works for UV-active compounds and is suitable for ionizable and non-ionizable compounds. However, for the potentiometric technique, the sample needs to be ionizable, but it doesn't need to be UV-active. The example shown in this slide is a deuterion. Deuterion compounds are not easy to determine, and results can depend on ionic strength. In the top graph, the green curve is the pKa determining water, and the orange curve is the pOKa. The pOKa is the apparent pKa determined in the presence of octanol. For log P measurements, the neutral species partition to the octanol layer, disturbing the equilibrium in the aqueous media. The more disturbed the equilibrium is, the more the pOKa shifts from the pKa curve, and therefore the higher log P will be. However, deuterion compounds are always charged, so they are not neutral at any pH. The only way to partition to the octanol layer is by forming ion pairs with the ion presence in the media. And this is what is happening in this example. The protonated species has a higher log P of 259 than the neutral charged species with a log P of 175. Like lipophilicity, two techniques can also be used for the determination of solubility, the spectrometric and the potentiometric technique. For the spectrometric technique, we are using again the check flash methodology, and for the potentiometric technique, the checksum methodology developed by Pion. Checksum not only determines the intrinsic solubility, it also tells you about the solubility of the metastable amorphous forms. This information is very valuable if you are seeking for a solubility enhancement strategy. The plot shown in this slide is benzoflumethiazide, an acid drug. With the checksum method, we obtain the graph of neutral species concentration with time. At the beginning of the experiment, the sample is in solution and ionized, and so the neutral species concentration is 0. Then the titrates are added, in this case is acid, and the neutral species start to increase as the titration approaches the sample pKa's. The neutral species increase until precipitation is detected, and the kinetic solubility is determined. After precipitation, the neutral species concentration decreases as the drug starts crystallizing. Eventually, it reaches the more stable form, the intrinsic solubility of the drug. Studying the supersaturation enables us to observe the extent, the maximum concentration of the neutral species in solution, and the duration of the supersaturation before the sample precipitates to the more stable form. In this slide, we have the example of isoxicam using plasdone as excipients, where we can see an increase of the duration of the supersaturation concentration with the plasdone K-type. However, in the case of dipyridamole, an increase of the extent of the supersaturation concentration is observed when the percentage of carbazole increases. We publish this data and the reference can be found in the slide. This was a very interesting experiment. We were performing a routine solubility assay of 1 hour duration, and the intrinsic solubility was determined at 400 micrograms per milliliter. The customer was quite surprised with the results, as their check-fast methodology gave them a solubility 10 times lower. We discussed this case, and we increased the duration of the experiments for 4 hours. The additional graph suggested that the first precipitation is an amorphous form with higher solubility, temporarily stabilized for 2 hours, and it takes 1 hour to convert to the more stable form, the crystalline form, with a final solubility of 45 micrograms per milliliter. This result was not only in agreement with the check-fast result from the customer, but it revealed an amorphous form 10 times more soluble than the crystalline form. In this slide, I've shown the applications for physicochemistry properties in discovery, preformulation, and formulation. Let's talk about dissolution. In this case study, the customer was interested in identifying the best salt for a new formulation. For this experiment, we used the Pion Inc platform. The vial is bigger than the Pion Series D3, so we added 40 milliliters of acetate phosphate buffer adjusted to pH 5. The sample was added as 3 millimeters compressed disc. The customer sent the API plus 4 different salts for a fully basic soluble drug. The goal was to identify the salt which reached the highest concentration in solution by the end of a 2 hours experiment. All the salts dissolved more than the free base, but a greater quantity was dissolved for the hydrochloride salt, which was selected for further evaluation. During the early stages of drug formulation, it's important to assess the performance of an API and the conditions relevant to the gastrointestinal tract. At Pion, we have a selection of low volumes dissolution assays to support formulations at biorelevant pH and with optional biorelevant media, FASIF and FESIF. Case study 6 shows the biorelevant dissolution of 3 APIs, phenethylene, indomethacin, and felodipine. 15 milliliters of FASIF and FESIF biorelevant media. This shows clearly both the effect of the media and the pH. FASIF and FESIF, illustrated by the red dissolution profiles, gives a significant increase in the dissolution performance of all 3 compounds. However, the medium pH, 6.5 for FASIF and 5 for FESIF in blue, have an influence on the performance of the ionizable compounds phenethylene and indomethacin, but not for felodipine. In this example, the customer wanted to quantify the difference in dissolution depending on the surface area exposure of the compound. The samples were prepared by pressing powder ibuprofen into 3 millimeter tablet disc. The left graph shows the mass dissolved after 2 hours using 3, 6, and 8 millimeter tablet discs. As expected, the more the area is exposed to the media, the greater mass release. The graph on the right shows the results of repeating the experiment, but in this case, using ibuprofen sodium salt. It's interesting to observe the effect of the salt, increasing the solubility 10 times for the 3 tablet sizes, in comparison with the free acid. The dissolution of 3 experiments reached plateau in less than 5 minutes. We have a lot of cases in analytical services where a biorelevant media conversion is requested. With the microdisc and informed instruments, we are able to include a dynamic change to the dissolution medium by the addition of a simulated fluid concentrate, allowing the conversion from a simulated gastric media to an intestinal one. The example in this slide compares the dissolution performance of an acidic API with the sodium and calcium salt. The effect of the switch to intestinal media from gastric is quite pronounced. Supersaturation, or the spring and parachute mechanism, is another important property for improving the bioavailability of poorly soluble drugs. Some APIs are capable of maintaining a temporary supersaturated state with higher solubility before precipitates and reaches equilibrium. This phenomenon can be assessed under controlled temperature and pH conditions using both the Pion Info and microdisc profiler. The extent and duration of supersaturated states can be manipulated by the inclusion of various excipients in formulations. The graph on the right shows a routine experiments performed for a customer. A solubility assay was previously performed to obtain the pH when the sample started to precipitate. The experiment was repeated. Please look at the API curve. And the assay was stopped at the pH just before the sample precipitates to observe how the sample will behave. After 40 minutes, nucleation occurred and precipitation happened very quickly, reaching the final value of intrinsic solubility. This example showed the opportunity of solubility enhancement. So Cavazole was used to enhance solubility, and the inclusion of Cavazole enables the API to remain supersaturated at the concentration of approximately 300 micrograms per milliliter for at least 3 hours. I would like to introduce the Pion SDI2, a surface dissolution imaging. The SDI2 provides high-resolution images of the samples as they dissolve. It's capable of capturing images at 2 wavelengths during the same assay, UV and visible. The UV data is used to calculate sample concentrations, so the drugs release. The visible light can be used to follow the physical changes that the sample experiences as it dissolves. 2 flow cells are used, the compact cell and the whole dosage flow cell. The compact flow cell is designed for powered samples. The whole dosage flow cell is based on a USB Type 4 flow cell design and is used for the dissolution of larger samples, like tablets or maybe capsules. In this slide, the compact flow cell was used to study the enhancement of solubility of dipyridamole using HPMC acetate succinate in different ratios as amorphous solid dispersion. The dissolution of the samples were determined using the UV light, and the images on the right-hand side show the dissolution videos at 10 minutes. The color images clearly show the dissolution enhancement of the excipients, as the dissolution curves captured from the same experiment. Increasing the proportion of HPMC in the dispersion increases the dissolution rate of dipyridamole. The SDI2 is able to combine measurements of drug release from whole dosages form, such as tablets or capsules, with monitoring a sample dimension, height and width, at the same time. Monitoring both parameters allow the to link specific physical events, for example, the disintegration of a tablet or the rupturing of a capsule, with the corresponding drug release. In the case of our example here, we can see that the absorption of water and swelling of the tablet allows the active ingredient to slow release over time, and continues to be released 9 hours after the experiment is started. If you would like to find out more information about SDI2 and its capabilities, please join us on the 10th of June for a full webinar dedicated to SDI2. In this slide, I've shown the applications for dissolution under pre-formulation and formulation. Let's talk about absorption and permeability. Understanding the dissolution characteristics of your candidates is very important, but it doesn't always tell you the whole story. It's important to probe the absorption characteristic alongside the dissolution to fully understand your samples. Pion has 24 years of experience in studying absorption across artificial membranes and a selection of services suitable for each step of the drug development. For discovery, we offer PAMPA, a high-throughput play-based assay to rank order for a large number of samples. The Pion microflux, a small volume absorption instrument, is used to assess the in vitro absorption potential in a small-scale media. Microflux can be used during pre-formulation stages to optimize prototype formulations or to assess potential excipients when sample materials may be scarce. The macroflux combines a flux chamber using Pion's PAMPA membrane, a GIT lipid with USB dissolution vessel. The in situ dissolution absorption measurements can be used to predict performance of a drug product, to compare formulations, compare batches, or to assess bioequivalence between generic and innovator products. Over the next few slides, I will describe some case studies to introduce these techniques. In the PAMPA technique, samples are loaded as a DMSO stock solution into the bottom donor plate containing target pH buffer. Placed on top is the acceptor plate, a 96 well plate with a PAMPA membrane in the bottom of the well. This forms the PAMPA sandwich, shown here with the donor chamber, membrane, and acceptor chamber. The membrane is loaded with GIT lipid to simulate absorption in the gastrointestinal tract. The acceptor well contains a pH 7.4 zinc buffer to simulate zinc conditions. The samples permeate across the membrane and the concentration in each well is analyzed. The amount of sample in each well, along with the sample retained by the membrane, can be calculated and a permeability value and rating given. PAMPA has been validated against human jejunal permeability and shows good correlation with in vivo results. In this slide is shown the example of carbamazepine. The top profiles show the dissolution and flux of the API alone. We see rapid supersaturation of the drug followed by a very rapid precipitation. When we add a solubility enhancer, Solu Plus, into the donor chamber, we can see that we get a much enhanced max concentration, and the concentration is sustained by the parachute effect, by the excipient for a number of hours. We can see in the flux graph that we achieve significantly greater concentration in the receiver chamber and a corresponding flux value of 2.25 times higher than the drug alone. The macroflux application is useful for predicting the bioequivalence of generic formulations with innovative products. Here we are comparing the flux performance of generic temilsartan formulations with the Boehringer Ingelheim Micardis branded formulation. The dissolution performance of the Actavis and Mylan formulations is observed to be behind the Micardis formulation, which is reflected in a lower flux, with flux ratios of 81.6 and 92.1% respectively. This predicts that Actavis formulation is on the borderline of the 80-125% bioequivalence acceptability criteria. When these products were brought to the public assessor, both were accepted, but with the comment that Actavis product was at the very borderline of acceptability. The macroflux was shown to be a very good predictor of bioequivalence. Case study 15 shows the dissolution study of adefovir and its co-crystals with the coformers resorcinol, orcinol, and hydroquinone. For this example, the Pion Inc system was used for the biphasic dissolution study, where the sample is dissolved in 4 pH sectors for 30 minutes each. The experiment is started in gastric conditions at pH 1.6. The pH changes to 4 in the second sector, and it's after the adjustment of this pH when the lipid layer, decanol, is added to mimic the absorption in the GI tract. Following sector 2, pH changes to 5.5 and 7.4. 2 UV probes are used for the quantification of the drug in the aqueous and the lipid layer. In the graph, we can see in the first sector the co-crystal orcinol with the yellow curve in aqueous media. It dissolves completely at the end of this sector. In the second sector is observed a reduction of the sample in solution. This is because the co-crystal is migrating to the decanol layer, the brown curve, dissolving a total amount of 7 milligrams. In this slide, I've shown the applications for absorption permeability in discovery, preformulation, and formulation. This is our Scissor system for subcutaneous injection site simulator. We use this instrument to predict the fate of biopharmaceutical post-injection into the subcutaneous space. The Scissor works by injecting a biopharmaceutical into a cartridge filled with our artificial extracellular matrix, ACM, which simulates the subcutaneous space in humans. The cartridge is suspended inside a chamber filled with simulated interstitial fluid. Over time, the drug diffuses down the cartridge and out by customized membranes into the interstitial fluid, indicating uptake on the drugs to the blood and lymphatic vessels. We can take aliquots of the interstitial fluid at time intervals for offline quantification of the drug release per unit of time. We maintain the pH of the interstitial fluid at 7.4 and keep the whole system at 34 degrees centigrade. We have an electrode which measures the pH and the injection site and we have 4 LEDs at different heights that read the transmission throughout the assay. From this data, we can infer any evidence of precipitation or aggregation events happening at the injection site. Here is a quick example showing the transmission data collected by the 4 individual LEDs. The cartridge at the start of the experiment is completely clear, meaning that all 4 LEDs show 100% transmission. Once the formulation is injected, if there is any precipitation or we have a suspension, this will alter the light between the LEDs and diodes, causing the transmission to decrease. In this example, we use an opaque formulation to visually demonstrate the transmission. Upon injection, the depot blocks channel 1 and 2. This is green and pink plot, causing a decrease in transmission. As the depot starts to migrate away from the injection site, channel 1 is no longer obscure and start to increase once more, green pot. And channel 3 start to be affected, blue plot. As the formulation migrates, channel 1 and 2 recover back to 100 transmission. Green and pink, and this is the green and pink curves. And channel 3 and 4 are then obscure, blue and orange. As the formulation starts to stay at the bottom of the cartridge, all the top 3 channels return to 100% transmission, and only channel 4, orange, is obscure. To date, we've worked with a range of samples types on Scissor, including antibodies, peptides, solid implants, such as acid pumps and polymeric rods, as well as in situ forming gels and small molecules. We are able to provide transmission data indicating precipitation events throughout the assays, and we can also quantify the drug release per unit of time throughout the assay time frame. Overall, Sciss is a great technique for excipient screening or rank ordering subcutaneous formulations, helping to better understand biopharmaceutical behavior post-injection. If you'd like to find out more information about Scissor and its capabilities, please join us on the 24th of June for a full webinar dedicated to Scissor. I hope this gave you a good overview about what we are doing in analytical services. For further information about anything I have mentioned today, or for details of the services I wasn't able to include in today's webinar, please visit our website. On the website, you can find our analytical service brochure. This brochure gives you detailed information about the techniques that we are using and the different analyses that we are offering. Additionally, you can contact your sales representative for any specific questions that you might have using the email address on screen now. We are also registered supplier of Scientist.com, an online marketplace for scientific services. All of the services discussed today can be purchased on this platform. Using Scientist.com can streamline the sample submission process as the Scientist.com Research Services Agreement, so can eliminate the need for individual legal agreement between supplier and customer. Quotations, purchase order, and invoicing are also all handled within the platform, making the process as simple as possible. All of the analyses shown in this webinar are possible thanks to the analytical service team. Here in UK, we have Imi, Ben, Emily, Sam, Andy, Luis, and myself. And based at our US facility, we have Oksana. And we have arrived to the last slide. Pion Analytical Service can support your projects throughout every step of the drug development journey with expert measurement and advice based on years of experience in the field. We offer spectrometric and potentiometric assays to determine physicochemical properties of the compounds. Small scale and companion dissolution analysis, dissolution imaging, controlled supersaturation, assessment of permeability and absorption with a range of techniques, consultancy projects to support drug formulation, bioequivalence studies, assay for biopharmaceutical with the Pion Scissor. Pion are licensed to work with controlled drugs in our UK lab. Please contact your sales representative if this service will be of interest to you. If you have any question, please send this over to us. We know that every sample is different and there is a story behind the compound behavior. We are happy to discuss with you the best technique or analysis to obtain the answers that you need. It was a pleasure to be here today. Thank you for listening.
Thank you, Rebecca. That was great. I think your team got a couple of questions throughout the presentation. So if you want to answer a couple of those, we have a few minutes.
Thanks, Karen. The Q&A window will continue to be open as we answer questions. So if you think of any that come up now that the presentation's finished, please continue to send them in and we'll do our best to get through as many of them as possible. Just to start off, Rebecca, I've had a question regarding sample submission. What's this process like? How would somebody go through the process of submitting a sample, and what information does Pion need in order to get the analysis started?
Okay, well, if you would like to submit a sample, the information that we need is we will send the sample submission form and needs to be completed. And the sample submission form is you can add all the information about your sample, your details of the company, and depending on the analysis, we will ask you for different properties of the sample, or if you have the structure, this will help us a lot to really design the better assay. Not only the sample submission form is asking for the sample and your details, is the toxicity of the sample. We need the MSDS sheets to check toxicology of your sample. And definitely we need this before the sample is sent to be able to tell you if we accept or not the sample. And yes, you will be in contact with the sales representative and you will get a quotation. So that means that then we need the purchase order and then we will start the sample with all this information.
Okay, thank you, Rebecca. This is almost a follow-up to that. It's just could you comment on the sample amount requirements for the services that you've talked about today? And she could also mention the turnaround time for our projects.
Yeah, okay. The quantity of sample is depending of the analysis and the number of analyses. For screening analysis, for example, we need only 1 milligram of solid or 200 microliters of 10 millimolar DMSO stock. This is for screening. For a gold or platinum standard assays is depending of the analysis that is requested, so could be between 10 and 100 milligrams. Maybe more or less is 10 milligrams for pKa and 5 more for log P. But if you are going for shake flask, then we are asking for more, 20 to 50 milligrams. And yes, and the consultancy or platinum, yes, the consultancy projects then is depending of the analysis that we are, but this will be discussed before the analysis started. And sorry, and the other question was? Andy? Andy?
Oh, apologies, left myself on mute. The other question was on the turnaround time for the submitted samples.
Yeah. Yes, when we have all the information that I mentioned before, that the sample submission for MSDS sheets and the purchase order and the sample, then it's 10 business days. From that, we offer a rush order service, and for that is 5 days or less. And if we have consultancy projects, we discuss this for the customer because it's depending of the amount of analysis that is requested.
Okay, thank you, Rebecca. We had a question come in during the presentation. I believe it was referring to Inform. I think I'll pass this one to Sam, if he's happy to answer this. How are the sample tablets added into the dissolution? And does the effect of disintegrated sample powder block the UV probe detection in any way?
Hello. Right. So yeah, the position of the tablet, this will change depending on whether we're doing IDR on the Inform or the microdis. So on Inform, the tablet die is suspended perpendicular to the base of the vial, level with the stirrer. I think Rebecca has a diagram of this on slide 22. Whereas on microdiscs, the tablet die is embedded in on microdiscs, it's embedded in a rotating disc at the base of the vial, similar to the Woods apparatus. With regards to disintegrated sample powder, in an ideal scenario, we would not see disintegrated sample powder from the disc, as then we lose control of the dissolving surface area. Though I'd add that in cases where we have turbidity in the medium, such as from disintegrated powder, our fiber optic systems have rather sophisticated baseline correction algorithms, in addition to second derivative data processing, which are able to correct spectral data for this. I hope that answers your question.
Okay, thank you, Sam. Next question that's come in is regarding the media that we offer for our services, particularly biorelevant fluids. And the question is, do you have any support for non-human biorelevant fluids, such as canine or feline gastric simulations? I think I'll open this up to either Sam or Carl if they have anything they'd like to add on that question.
With non-human biorelevant fluids, among other media, our standard array of AS assays is more or less restricted to a selection of sort of standard media, buffers and fastive and festive and whatnot, though I would add that our instruments are capable of handling quite a wide array of other media. So while we don't offer these on our standard AS assays, usually do get in contact with us if there is anything that you would like us to try and work with, and we can work through these sorts of things on a case-by-case basis.
Okay, thanks, Sam. That's also covered another question we had referring media in general. So yeah, just to reiterate what Sam said, if you do have a specific media for dissolution or absorption measurements that you would like us to work with, please discuss that with your Pion sales representative. We are capable of working with the majority of dissolution media that you may wish to work with, and we're always happy to have those conversations. Rebecca, during your presentation, you discussed the different methods we have for determining the log P or log D of submitted samples. What are the measurement or measurable ranges of these techniques?
For potentiometric, okay. For potentiometric, we could determine log P's from -1 to 5. However, it depending a lot of the sample pKa, because here we are limited by the pH range. So if we have a high acidic pKa, which normally shifts up with the presence of octanol, if the log P is so high, we are not going to able to determine. However, for spectrometric technique, we are using the check flash technique, and the ranges are between -2 and 4.
Thank you, Rebecca. Finally, we've had something in regarding scientist.com, which you mentioned right at the very end of your presentation, referring to how that process differs from the standard sample submission process. I think I'll take this one. So you would need to, in order to submit through scientist.com, you would need to sign up on their website. It's a fairly simple process. And then you would submit a request for services directly to us, and that would come straight through to us here. We would then be able to review your request, ask any questions that we might have to make sure that we get the analysis right for you, and then we would submit a scope of work to you, which is a proposal of the work that we would perform along with a quotation. And then that can be purchased directly on the platform, and POs generated through the platform as well. So it can help in some circumstances to just streamline the submission process a little bit. But you would need to be a member of the website before you can submit. Okay, so I think that's covered the questions that we've had in. I don't know if there's anything that either Rebecca or Karen you'd like to add just at the very end of the webinar here.
Yeah, so for any questions that you will have maybe later, when we do have this recording, and if you think, oh, I would like to have more information about any of the techniques that we were explaining today, please, yes, contact us. We will answer all your questions.
Yeah, thank you, Rebecca. I just wanted to thank everybody for joining us. And as I mentioned, you'll get the link with the recording and questions and answers as well. That initial email will come from Ring Central, but it will include my email too. So as Rebecca mentioned, we're happy to answer any questions, and your Pion contact will also just be contacting you to make sure you were able to get the link and the file's too big and that type of thing. So thank you, everyone. And it will also include a link to upcoming webinars as well. So we appreciate everybody's time. Thank you so much.
Thank you.